First of all, I wanted to thank all the ERGA team that is working on the sequencing of the selected species. We are very grateful to be able to participate in this amazing project. And that being said, I'm Pablo Aguado-Ramsay, a PhD student at Autonomous University of Madrid, Spain. And my thesis attempts to understand the actual diversity and evolutionary history of several pseudocryptic species of mosses. So in this context, we presented several species, out of which 2 were selected: Orthotrichum diaphanum, highlighted in orange, and Lewinskya acuminata, highlighted in green. The former, Orthotrichum diaphanum, is widespread in the Northern Hemisphere, particularly in Europe, North Africa, and North America. However, in the Southern Hemisphere, distinct populations are known from the Northern Andes and South Africa. It is an easily recognized species in the field because of its characteristic long hyaline hairs on the leaves. And the later, Lewinskya acuminata, is also widespread through the Mediterranean basin, with disjoint populations in California and Ethiopia. It is also very easily recognized by its long acuminate leaves and its characteristic fusiform capsules with reduced peristome and punctured mouth when dry. So these mosses are 2 of the most representative species in the epiphyte communities here in Europe, and they're not difficult to find and collect. However, as you see at the left, mosses are small and they can live intermixed. So I spend a lot of time, basically a week per species, gathering all the material that was needed for sequencing, carefully selecting each individual gametophyte to prevent cross-contamination as possible. So back again, these 2 species are from the Orthotrichaceae family, which is the first most diverse family among bryophytes, comprising around 800 species. Recent studies on this family show that it has a complex evolutionary history, and there may be a greater diversity than previously expected. So our team is actively working on several pseudocryptic species complexes, as is my doctoral thesis. So to this end, we're applying integrative taxonomy with modern massive sequencing techniques like genotyping by sequencing and HypeSeq. So having a relatively close reference genome will facilitate genome assembly and many other analysis. So it will allow us to delve into the complex evolutionary history of this group of bryophytes. And if you have any questions or you would like to find more information, you can visit our website, ortho3.net. Thank you very much.